Oh, oh yes. Okay. All right. Now we're going to discuss translation, the last step in the last step in gene expression or protein synthesis. Look at protein structure and the initiation, elongation, and termination of translation. And then lastly, we'll summarize it. So the primary structure of proteins is the amino acid sequence, and that's what we're determining here with translation. Secondary structure is the interaction of non-adjacent amino acids. Tertiary structure is the three-dimensional conformation, or how the entire polypeptide chain folds up. And then quaternary structure is the number of polypeptide subunits in the functional protein, monomer for 1, dimer for 2, trimer 3, etc. And this shows you each of those. And so it's primary structure that is being determined here with translation, although the primary structure does also determine the secondary and tertiary structure. So amino acids are linked together by these peptide bonds where the amino group of 1 covalently bonds to the carboxyl group of the adjacent amino acid. All peptide bonds are the same regardless of which 2 amino acids they're linking together. So here's the typical amino acid formula with the amino group on the left and the carboxyl group on the right, and then sticking down is the R group. So it's the R group that differs between the 20 different amino acids. The rest of it is the same. So this shows you hydrophobic or non-polar amino acids. So it's the R groups that are sticking down below the green boxes. The little green boxes are the same between each amino acid. And R groups can be very simple, like in glycine, it's just a hydrogen, or alanine, it's just a methyl group, to pretty complex, like a double ring structure in tryptophan. The rest of the amino acids are shown here. Some with hydrophilic or polar side groups, others are acidic or basic. Basic ones tend to have nitrogen in the R group, and carboxylic acids tend to be in the R group of the acidic ones. Obviously, you don't have to know all these amino acids, but just know the basic structure, i.e. what's in the green box. This shows the formation of a peptide bond with 2 amino acids coming together. And so about the only chemical bond I want you to know in this class is the peptide bond, that thing in the little purple box on the right. Now the secondary structure are hydrogen bonds between non-adjacent amino acids that are fairly close to each other in the polypeptide chain. 2 types are recognized: an alpha helix, a right-handed coil, and a beta sheet, sort of when the chain folds back on itself. And the alpha helix is on top here, the beta sheet on the bottom. There's another way of looking at it. The beta sheet kind of reminds me of one of those temporary room dividers. Then tertiary structure is the 3-dimensional conformation of the folding of the entire polypeptide, and that results from the interactions of R groups. For instance, cysteine residues can form disulfide linkages. Hydrophilic groups cause the thing to fold in such a way that they're on the surface, and hydrophobic R groups do the opposite. And so this shows you some of the ways that the R groups can interact, causing the whole polypeptide to fold in a certain 3-dimensional way. And then quaternary structure is merely the number of polypeptide subunits in the functional protein. So here in hemoglobin, it's a tetramer. So there's 2 beta chains, which come from chromosome 11, and 2 alpha chains, and the gene for that's on chromosome 16. And so a functional hemoglobin molecule has to have all 4 polypeptides. So this kind of summarizes translation visually, with our DNA at the top giving rise to 3 different types of RNAs: the ribosomal RNAs, the messenger RNA, and the transfer RNA. And then those things combine with the ribosomal subunits, and then we can get translation to occur. So here, this compares the prokaryotic ribosome at the top with the eukaryotic mammalian ribosome down at the bottom. So of course the eukaryotic ones are larger and have more molecules involved. 3 ribosomal RNAs for the small, for the large subunit, and eukaryotes for only 2, and the large subunit for the prokaryotes, and 1 each for the small subunit. So then, in translation, of course, the ribosome will interact with the messenger RNA starting at the initiation codon AUG and continue on down the messenger RNA molecule until it reaches a stop codon. So, in eukaryotes, it occurs in the cytoplasm. Of course, involves ribosomes. There's an initiation phase, an elongation phase, and a termination phase, just like with nucleic acid synthesis. However, these are more complicated, involving a lot more molecules. So, a polysome or a polyribosome is found in prokaryotes, where transcription and translation can occur simultaneously. We see in the diagram up at the top there the RNA snaking out of the transcription bubble there, and before it's completely made, ribosomes are clamping onto it and beginning to translate the mRNA into polypeptides, as shown by those little chains of purple balls, and a photomicrograph of it is occurring down below. So here's a close-up. So the RNA polymerase is cruising down the DNA, making RNA, and before the RNA is fully made, translation is already occurring. So, and remember, that can happen in eukaryotes because, of course, transcription and translation occur in different parts of the cell. The first things that has to happen is charging the tRNA. A charged transfer RNA is a transfer RNA with its amino acid attached to it. So enzymes do this. Enzymes called aminoacyl tRNA synthetases, and they're named for the amino acid they attach. So, for instance, leucyl tRNA synthetase attaches leucine to its correct amino acid. To its correct transfer RNA. Prokaryotes have one set of 20 aminoacyl tRNA synthetases, of course, one enzyme for each amino acid. The eukaryotes have at least 2 sets, one for cytoplasmic and then one for mitochondrial transfer RNAs. So this shows you a model of tRNA on the left there, and remember, it's the 3' end with ACC where the amino acid attaches, and it's sort of blown up there how the amino acid attaches. It's the carboxyl end that attaches to the adenine of the transfer RNA. And here's the process, and it requires ATP, so energy is required to charge the transfer RNA. Now, let's look at transfer RNA binding sites in the ribosome. There are 3. They're at the junctions of the 2 ribosomal subunits. Only 3 tRNAs can fit in the ribosome. The site with the growing polypeptide is called the P site, P for peptidyl. The aminoacyl site is called the A site. That's where the tRNA with the new amino acid comes in. And then the exit site is the tRNA that's already given up its amino acid and it's ready to be ejected from the ribosome. So here's what it looks like. So the P site is in the middle. E site to the left there. Notice that tRNA doesn't have its amino acid anymore. It's no longer charged. And then the latest one that came in carries the amino acid alanine. And in here, in this diagram, the messenger RNA is the red line through the middle. The large ribosomal subunit is on the bottom, the smaller one on the top. So the codon in the A side of the ribosome is said to be the reading frame. And the frame shift is a mutation where a shift in the reading frame occurs. This can be a substantial mutation. For instance, this is 4 CAU codons that code for 4 histidine amino acids, but if you have a frame shift, just one base over, then you get 4 AUC codons that give you 4 isoleucine molecules instead, and so that can really change the protein. So that's what a frame shift is. So AUG remembers the initiation codon, codes for the amino acid methionine. Now in prokaryotes, it codes for a special form of methionine called N-formal methionine, but regular methionine is found in other AUG codons that aren't at the beginning of the mRNA sequence. And the N-form methionine tRNA is the only charge transfer RNA that can bind at the P site. Eukaryotes, we have a special tRNA for the initiation codon, but the methionine is the same as any other methionine amino acid. So the first step in translation is the formation of the initiation complex. So step one, so we have these initiation factors, which are proteins that guide this or help this process. Initiation factor 3 binds with the small ribosomal subunit, the messenger RNA, the first transfer RNA, and initiation factor 2 as well. And in the second step of this process, the step 1 complexes combine with each other with initiation factor 1 and with GTP, forming the 30S initiation complex. That's what IC stands for, initiation complex. The GTP is an energy providing molecule like ATP. Then the last step here, GTP, of course, is cleaved to provide the energy. Initiation factor 3 is released, and the large subunit, the 50S subunits, joins the initiation complex, and the other initiation factors 1 and 2 are released as well. Now, in us eukaryotes, we have 11 initiation factors, so things are much more complex in us eukaryotes. So this kind of shows you, here's our, at the top, our 2 ribosomal subunits, then the 30S ribosome initiation complex there in C, and then the entire initiation complex down at the bottom there in D with only a single first transfer RNA, and now the reading frame with the codon UGC is open, ready for the next tRNA to come in. So this shows our, again, our 3 tRNA binding sites in the ribosome. Now, how does the ribosome recognize the messenger RNA molecule? Well, in prokaryotes, there's it's called the Shine-Dalgarno sequence, just upstream from the AUG, the start codon, and it's complementary to a sequence in the 16S ribosomal RNA molecule, which is part of the ribosome. In eukaryotes, we don't have this, but the ribosome recognizes the messenger RNA due to the 5' cap, where initiation factors bind. Also, the poly A tail bends around and attaches to ribosomal proteins, and the scanning hypothesis states that the ribosome then scans for the messenger RNA for the start codon. The start codon is embedded in something called the Kozak sequence, which reads 5' to 3'. You can see there ACC, then AUG, the start codon, and another G. So here are Shine-Dalgarno consensus sequences, and then you can see at the bottom there, it's that sequence in yellow there. That's UAA, GGA, GGU, complementary to that 16S ribosomal RNA in the small ribosomal subunit. And then you can see just to the right of it is the start codon, the AUG codon. This shows the process in eukaryotes, where we've got the ribosome there and these cap binding proteins that attach to the 5' UTR in the ribosome, and then the poly A tail bends around with poly A proteins attaching to the cap binding proteins. And then the dark green area to the right there is the area that will actually be translated into protein. You can see AUG start codon, UAA is a stop codon. So here's that Kozak sequence that has the start codon embedded in it, and the scanning hypothesis states that the ribosome scans for this sequence in the messenger RNA. Now, elongation in translation is a 3-step process. First step is attachment of the new tRNA, then the formation of the peptide bond between the adjacent amino acids, and then translocation, which is the movement of the ribosome along the messenger RNA from the 5' to 3' direction. So I'll have to discuss what happens in each of these. So for a new transfer RNA to bind, we need 2 elongation factors. And it hydrogen bonds at the A site or the reading frame in the ribosome. Elongation factor EFTU binds to the new transfer RNA. Then elongation factor TS binds the GTP to the other elongation factor so that GTP can be hydrolyzed, providing energy here. It can happen pretty quickly in prokaryotes. 20 amino acids per second can be incorporated into a growing polypeptide. And in us eukaryotes, we have eukaryotic elongation factors 1 and eukaryotic elongation factor 2 that do similar functions. So this kind of shows you here. So here we have our first tRNA in the left at the P site. The A site is open, and now comes our second transfer RNA that is charged with the amino acid glycine. And you can see the little pink guys are the elongation factors, and the little yellow thing is the GTP, which gets hydrolyzed, providing energy for this process. And now the ribosome, shown on the right, has the new tRNA hydrogen bonded at the A site or in the reading frame there. So second step then is the formation of the peptide bond between the two amino acids. And there's an enzymatic center of the large ribosomal subunit that does this called peptidyltransferase. So the carboxyl or the acidic end of the amino acid at the P site and the amino end of the amino acid at the A site will form the peptide bond. And we see that. And that happens as the ribosome is moving. So now we have a dipeptide where the formal methionine has bound to the glycine. And now that last amino acid, last tRNA that came in is now in the middle of the P site with two amino acids stuck to it. And the first tRNA has been ejected from the exit site. Translocation then. Elongation factor G, or also known enzymatically as translocase, is involved in this. And the tRNA at the old P site is now moved into the E or exit site. In eukaryotes, eukaryotic elongation factor 2 does this. And so we can see here, over to the right there, the ribosome has moved. And now we've got a new open A site, and the E site has just had the first tRNA ejected from it. This shows an actual photomicrograph of translation in action. So the larger balls, dark blobs in the middle of the thing are the ribosomes, and the little black arrows are pointing to polypeptides that are actually being made, sort of sneaking out of the ribosomes. And then, of course, the molecule that's running through the ribosomes is the messenger RNA. Now, termination. So when a stop codon appears in the A site, a protein called a release factor binds the A site instead of the transfer RNA molecule. And this release factor causes the polypeptide chain to be released. And of course, there are 3 release factors. Release factor 1 recognizes 2 of the stop codons, UAA and UAG. Release factor 2 recognizes UAA as well, and UGA as well. Release factor 3 then causes the messenger RNA to be released, so it does not bind at the A site. And then initiation factor 3 binds to the 30S ribosomal subunit that's aids in dissociating, aids in the complex kind of coming apart, if you will. So here we have our UAG stop codon in the A site there. And so release factor 1 or 2 will bind there, and then release factor 3 is going to bind to the ribosome. As we can see here, release factor 3 using GTP to provide energy. And now the whole complex falls apart, and the polypeptide is released. And this shows you termination from start to finish. So first, the polypeptide is released as one of the other RF1 or 2 bind to the A site. So sort of reviewing the whole process then, here's a charge transfer RNA. It's amino acid, our messenger RNA with the start and stop codons, our large and small ribosomal subunits. And of course, this happens in the cytoplasm of eukaryotic and prokaryotic cells. So here's tRNA charging at the top, followed by initiation under that, then elongation below that, then termination, and then peptide release at the bottom. So summarizing this process, it's fairly energetically expensive. It costs at least 2 ATP and 2 GTP molecules per peptide bond. So in the bacterium E. coli, about 90% of the energy used is for translation. So it costs a lot of energy to make those proteins. 300 amino acids is pretty much a typical polypeptide. A prokaryote can make one of those in about 20 seconds. The eukaryotes take about 2.5 minutes to do so, a little longer. And this table shows all the molecules associated with or that are involved in translation and what they do. So if you can tell me all this, you'll be golden on an exam. So let's review some things by the numbers. The central dogma really by the numbers, there's only 4 bases in the nucleic acids, A, C, G, T in DNA, A, C, G, U in RNA. Only 3 really important RNAs for translation, mRNA, ribosomal RNA, tRNA. Genetic code is 64 codons. 61 code for our amino acids, 3 are stop codons. And there's only 20 amino acids. Who'd have thought that life would boil down to such really a small number of different molecules? I think that's quite interesting. And that's the end of translation.